Back in the first few lectures, we distinguished between two different computational problems that we need to solve when analyzing sequencing data. So both of these problems arise when we want to take a collection, a large set of sequencing reads, and figure out where each of them came from. Which you can think of these reads as being like puzzle pieces, and you can think of the problem we're trying to solve as being like putting together a puzzle. We just spent a while discussing the first version of the problem called read alignment, and in read alignment it's analogous to the situation where we're trying to put together a puzzle but with the help of the picture of the completed puzzle from the lid of the puzzle box. So the picture of the completed puzzle is the reference genome that we would use, such as the human reference genome, for example, and that sequence came from the Human Genome Project. Now we're going to discuss another version of the problem called assembly for short, but it also goes by longer names such as de novo assembly or de novo shotgun assembly. De novo just means from scratch, and the shotgun just refers to the fact that the reads are coming randomly from all over the genome. So in this version of the problem, we do not have the benefit of being able to see the picture of the completed puzzle. So we might be studying a species, for example, that's never been sequenced before. In fact, when the Human Genome Project was conducted, it was the first time a human genome was being completely sequenced. So that project had to solve the de novo assembly problem with respect to the human genome. That was a very, very big problem, and as we'll see, this problem is generally a fundamentally hard problem. So it's definitely more computationally work-intensive than the corresponding read alignment problem, but it's also just fundamentally difficult. Some of the things that make it hard are things that we can sidestep or we can deal with, but some of the things that make it hard are things we just can't avoid. So we're going to have to live with certain unfortunate realities. But that said, we will also encounter a few really good ideas and some very profound ideas that form the basis of our modern tools for solving these problems. And furthermore, there's reason to hope that the assembly problem is one that technology will help us get even better at in the future. So we'll start to tackle this problem in the following lecture.